All right, let me get started, okay? And thanks for everyone for joining us today, as well as on Zoom. So I'm Sean Palate. I'm a Ph.D. student in the RNA-sequencing lab in the School of Dentistry, and I have a great pleasure and honor to introduce Dr. Jeremie and Oliver Piña today for this month's SCSAP Seminar Speaker Series. So Dr. Piña is an impassioned clinician engineer, consultant, and entrepreneur with particular interest in cleft palate and craniofacial health. He earned his Ph.D. in biomedical engineering from the University of Utah in partnership with the NICHD, and is currently a third-year dental student at the University of Maryland School of Dentistry. Jeremie has also earned bachelor's degrees in microbiology and biomedical science from the University of Hawaii at Manoa and Young University, respectively. It doesn't end there. During his PhD training, he also completed a Master's of Science degree in Healthcare Delivery Science from the Mayo Clinic and Arizona State University, and a Master's of Business, Healthcare, or Business Administration from the University of Utah, David Eccles School of Business. His scholarly work has largely focused on the single-cell spatial omics of murine palatal development, and has been published extensively in high-impact journals such as in Nature Communications, Journal of Dental Research, and Tissue Engineering, and has amassed over 800 citations across 86 peer-reviewed papers that Jeremie has published so far, in clinical, translational, and basic science journals as well. His work has also been recognized by a myriad of awards, and not having time to discuss all of them, I'll highlight just a few that he got this year alone. So first, they're all first place. First, in the International Association of Dental Research and American Association of Dental Oral and Craniofacial Research patent competition. For those of you who are not aware, this is sort of the highest award that a dental student trainee can get for basic science and dental research. He also won first place for a podium presentation at the University of Maryland Graduate Research Conference and an elevator presentation at the 13th Structural Birth Defects meeting. His scientific accomplishments are capped off with a vested commitment to mentoring and academic service, having trained numerous undergraduate trainees, as well as serving on executive boards of the American Association of Dental Oral and Craniofacial Research, as well as the American Council of Academic Plastic Surgeons and Biomedical Engineering Society. So his ultimate goal as a clinician, scientist, and surgeon, bioengineer, basically, is to have a research program that's focused on craniofacial anomalies to better translate novel treatments from the bench to the bedside. So he's undoubtedly a rising star in oral health, and it's really a true honor to introduce him today. And without any further ado, Jeremie.
Thank you, Sean. Wow, that was way too many words you spent on me, Sean. Thank you for for all that effort, and thank you all for joining me and for allowing me to join you here in Ann Arbor. It's my first time at the University of Michigan, and I hear all of the many good things that are touted around the academic pipelines about Michigan. So I hear it's all true. It's a great place, and I'm seeing it firsthand. So thank you for allowing me to come and present some work. So this is kind of the gist of my talk today. It'll touch on a few different new approaches that we've taken in the field to try to deconvolute the molecular biology of cleft palate. That's really kind of the resounding message here, is that there's a lot that we have yet to discover about the molecules that may be causing this global, burdensome disease that affects so many people worldwide. And that's a QR code to my contact info if you ever want to get in touch with me. Let me make sure this clicker is working. Yes, it is. So I understand that this room may not be full of people that really study the same thing that we do. Sean and I are kind of in the same vein, but I actually love that this program here at the University of Michigan brings together people all across the campus, it seems, from all different disciplines. So this is just a plug for the type of work that we do, that really our face and the structures underneath are really such a core part of who we are as human beings, such a big part of our identity. And if one, small anomaly may creep up during development or as a result of cancer or trauma, that can really impact the trajectory of your life. And I think that that's something that we can all at least relate with, even if we don't study this particular process. But, being a clinician at heart and, a scientist by trade, I really try to keep all of our actions in the lab clinically relevant. So this is really my North Star, just remembering which patients we're trying to reach and serve through all of our work in the lab. And so this is just some information about the prevalence and the incidence of cleft palate. It is probably more common than a lot of people appreciate. You know, about 1 in 700 live births on average around the world, depending on the region and the different environmental exposures. But really, this is an issue worth trying to address because the burden is so high, you know, physically, mentally, psychosocially, you know, financially. So this is really a big deal that I think that clinically we're coming up a little bit short. And this is just a snapshot of the whole timeline, not even gathering all of the different milestones, but these are the major ones that we reviewed in one of our articles, just to give you an idea of how many steps these patients must go through in order to just live some sort of semblance of a normal life. You know, there are multiple interventions at multiple different time points and a lot of complications that I personally saw in the clinic as a student and realized that I needed to go back to the bench and really understand things at a deeper level to be able to try to change our clinical dogmas. And this is exactly what we started to realize when I sat down with my PI, just how many gaps remain still in our understanding of the different molecules that could be allowing that palate to form properly in a developing embryo. And in particular, what is the spatiotemporal profile of many of these known genes that may be implicated in this process, but where are they expressed? How are they expressed? At what time point are they significantly playing a biological function, which allows for that palate to form and function properly? And perhaps this lack of knowledge is what has put us in this precarious clinical situation where we have a very limited scope of preventive or corrective therapies to offer these patients. So that's really kind of the premise of where my PhD began. And we wanted to kind of bring ourselves back to the fundamental principles or the fundamental just basics of how our palate is forming in the first place. So this is just kind of a whirlwind of our Nature Communications paper. You can read the whole thing if you follow that QR code, if you're really impassioned by it, but I'll give you the high points as we go through. But our first step was to really understand which are the genes from a bird's eye view, from just a bulk tissue level, that are active, and at which time points are they active in a normal palate situation. So which genes are crucially important for that palate to go from two, you know, flanked bilateral shelves and to be able to fuse together properly and give you that roof of mouth that is essential for your speech, for your swallowing, for your daily function. And to do this, we, you know, microdissected out the palate. You can kind of see on that far left image, or yeah, your left. I microdissected out using, you know, high-grade microsurgical instruments and my dental school loops to see each of these palatal shelves, and did that for each of these time points during development, and then ran bulk RNA sequencing on, you know, biological replicates of each of these stages in development, and constructed this library that had this temporal sequence of gene expression. And then we compared each of the adjacent stages of development against each other to try to see which genes were going up and down in each of these phases of development. And this is like the initial snapshot of these 5 different phases of development in a principal component analysis. So it's really just showing us transcriptional similarity between these different data points. So 3 different biological replicates of palatal shelves from each of these stages in time. It gives us this pretty clean break between a crucial moment in the palate's morphological process when these 2 bilateral shelves come together and they fuse, or the epithelium really integrates and allows for the mesenchyme to form that, you know, solid bridge of the roof of the mouth. We term that event fusion. The 2 stages prior to the fusion event are clustered off to the left, and all of the stages after the fusion event are clustered off to the right. So that kind of indicates that fusion itself may be, you know, defining this transcriptional event where something, you know, significant is happening, where there's a big change in gene expression. So we wanted to like delve into that particular phase in development. And what we found was our differential expression just lit up like a Christmas tree. You know, both upregulated and downregulated genes were highly differentially expressed. And on functional enrichment analysis, these plots on the right, they're kind of messy, but the main take-home points were that a couple of key biological processes were lighting up as significantly activated during this time point in development. One was bone formation, and the other one was ciliary function. And so we really saw that these processes only lit up at this time point from E14 to E15.5 at the fusion event of the palate. So we wanted to try to see what this looked like in the tissue itself. So we wanted to validate this bulk RNA sequencing finding. So we took this to the single cell level. We ran a multi-omic single cell analysis where we looked at RNA expression as well as ATAC chromatin accessibility in these cells. And we identified that the osteoblast cells or the bone forming cells of the palate only appeared post-fusion, which means that this fusion event really preceded and coincided with this osteogenic differentiation of these palatal mesenchymal cells. So this was an important validation exercise to show that our bulk RNA sequencing findings were legitimate. And then we wanted to see it in situ. This is really what we love as a histology-based lab, and as developmental biologists, we want to see where these genes are lighting up specifically in time and space. And so you can really see that stark difference between that pre-fusion, the E14.5 to the post-fusion E15.5, and several of these markers. It's really qualitatively you can really see the stark increase in expression, and each of these are bone cell markers at different phases of osteogenesis. RUNX2 at the pre-osteoblast stage, COL1A1 a bit more diffuse expressing, but common in the osteoblast, the functional osteoblast, and then osteonectin or SPARC is a required mineral for extracellular matrix mineralization during osteoblast differentiation. And then SOST, the terminally differentiated bone cell marker, also only appeared at E15.5, kind of signaling that bone cells had reached their peak in differentiation status at this phase. So this was an important finding that we felt like we had confirmed in bulk and single cell level. But we weren't quite satisfied with kind of the limited number of genetic hits that we could analyze in situ. So this was kind of in the early phase of spatial RNA sequencing, when we had the Visium manual assay at our fingertips, which had these, if anyone isn't familiar, I think you are, if you're in this lecture hall, you're probably quite familiar, but this is back when, these barcodes were a bit spaced apart. So we're not quite getting this single cell resolution, but you are able to sequence in space the entire, tissue context that you're placing on top of that glass slide. So what we did is that we really focused on these two time points, the pre-fusion and post-fusion, E14 and E15.5 mouse embryo. And we were able to manually select the barcodes that overlaid the palate. So in this next slide, you can see in the red overlay, I manually selected out the palate barcodes. And then we were able to do a differential expression analysis of these spatially resolved barcodes within the palate. And this gave us, of course, like a much broader viewpoint than we could capture using RNA scope probes until we die. So this was an important phase for us to then compare to our bulk RNA sequencing findings and kind of use this as an integrated approach to revalidate our findings. And that's exactly what we were able to do here. We took the top 5 differentially expressed genes from our spatial RNA sequencing of the palate. And then we mapped them in that bottom left corner plot. That's from our bulk RNA sequencing. So it shows that same trajectory of increase from E14 to E15.5 in all 5 of those genes. And 3 of those genes had never been discussed in the palate previously. So this was kind of our unexpected finding that these genes actually were likely playing an important functional role, and all of them, upon further analysis and reading in the literature, because I wasn't an expert in these genes, I'm still not, but they all have something to do with cilia function. And so that seems to also map back to our bulk RNA sequencing finding that osteoblast differentiation and cilia function were lighting up at the phase of palate fusion. And so this is just kind of a schematic representation of these 3 novel genes that were known elsewhere in the body, but not described in the palate previously. This is where they are expressed in spatial orientation. And then, not quite satisfied with that barcode output, we wanted to look at these at, single molecule punctate resolution. So this is an RNA-seq assay where we looked at, these different genes, and we found that a couple of them, DEUP1 and LRRC23 in particular, were quite nicely expressed within alkaline phosphatase positive, palatal osteoblasts, meaning that, we'd have to do some more, reductive, functional assays to really deduce their exact role in, palatal ossification. But it seems that there is some activity, coinciding with osteoblast differentiation with these novel cilia genes. So this is just kind of a summary of our key, salient findings, that this was a kind of a unique, multimodal data set of the normal palate as it develops. We're able to identify some novel gene hits that hopefully can lead to some new, regulatory networks of discovery and some new preclinical models to study the palate as it forms. But, of course, we're driven by, we're trying to understand normal development always. Of course, there's always more to discover, but we're really driven by the abnormal. And this is what we want to try to, reach patients at some level, at some point after all of this work. So, I've shown you a lot of interesting, intriguing data of what normal palate looks like at a transcriptional level. But really what we're trying to do now is compare this kind of ground truth of this foundational framework to a cleft palate model, which was really our next step, was to then hone in on a cleft palate model that we'd studied previously, but never really with the tools that we have available nowadays to really delve into like the nitty gritty of spatial biology. And so here, this is kind of the link to the next phase of this project that was recently published in JDR, Journal of Dental Research, where we really combine like a multi-omics approach to study the cleft palate, spatial biology as compared to our wild type framework that we just analyzed. And so our first step was to actually identify the transcription factor PAX9, which is a known transcription factor that orchestrates Wnt signaling within the entire body. And there's been a lot of foundational work done by my PI, Dr. Rina D'Souza, and others as well on this transcription factor. And it is, crucial for axial patterning. It's crucial for limb bud formation, and it's also crucial for the palate to form. So when we remove PAX9, you get a fully penetrant secondary cleft palate. So it's a great model to study, this particular tissue, because you're able to 100% of the time recapitulate your disease model of interest. And something that we hadn't really known, or no one had really identified, is that PAX9 in particular has a very unique, signaling milieu within the mesenchyme. And you can see it kind of clustered off to the right, both in RNA expression and in ATAC accessibility, that it is distinct from the other mesenchymal cell populations, this PAX9 hub, which may point to its regulatory niche, exerting on other cells within the mesenchyme and even the epithelium. And when we take away PAX9, we can kind of see some striking, phenotypic and genetic differences. So the first is, in the upper left, the micro CT, you can see that the bone really fails to form completely towards the axial midline. And so that's obviously, that's the manifestation of the cleft palate. But we haven't really understood what the role of PAX9 and its associated signaling networks could be on bone formation in particular. We can kind of, make some judgments of, qualitatively of the bone matrix being a bit more compressed, a bit more spatially disoriented than in the wild type. But we wanted to try to understand which of the known molecules that have a relationship with PAX9 may be impacting that bone from forming properly. And so we looked at a couple of molecules which we had studied previously in the lab, and found that PAX9 actually binds directly to these antagonists of Wnt signaling to help propagate Wnt signaling when it's present. When it's absent, DKK1 and DKK2 both upregulate significantly within the palate. And so we're seeing that in these single cell RNA sequencing outputs. And we're seeing that it's not necessarily an increase, or it's not necessarily a decrease in bone cells that we're seeing in the PAX9 knockout. It's more it's actually less differentiated bone cells that we're seeing. So you can kind of see in this proportion chart that, early and late osteoprogenitor cells, they don't, you might look at that bone and think that there is less bone formed there, but really we're starting to realize that the number of cells may not be less. It may just be that they are less differentiated or less spatially tuned or patterned. And PAX9 as a patterning transcription factor could be playing a role in helping that bone find its way in the palate as it develops. But in order to really delve into that, we needed to see this in the tissue itself rather than make judgments from our single cell RNA sequencing. So this is kind of a progression of our technologies that we were using at the NIH, and we landed on this Xenium in situ tool, which we felt was the proper way to analyze this at a high enough resolution, but also cover the breadth of targets that we needed to best analyze and answer this question. So, you're probably quite familiar with this workflow, but this is FFPE tissue, still, two-dimensional histology slices, but you have a bit more area to work with compared to the Visium, whole transcriptome sequencing. And it's not a barcode based, it's a probe based, analyzer. It's an imaging based assay where you are, more similar to an RNA scope or traditional in situ hybridization, but as it uses a technology called rolling circle amplification, you are able to, you don't have the issue of optical crowding that you do when you're looking at, traditional immunofluorescence. So you're able to pool many probes within the same tissue. And so at the time, we were kind of early adopters in our space at the NIH. And so at the time, they allowed us to build a custom probe set of 350 genes that could cover a lot of the different clusters that we identified in our single cell RNA sequencing from prior studies. So we custom designed out all of these, signaling cascades and cell types that we wanted to analyze within the palate, and many others I couldn't cram onto the slide, but this was kind of the way that we designed our, in situ workflow, was to pull from our single cell RNA sequencing data that we knew very well at that point to kind of customize our own, custom cocktail of probes. And then, just like any, histology-based assay, you slice your tissue and you place it, and then you're able to see all of this in situ, and then you can stain with H&E for morphology. So we did this for both wild type and our PAX9 knockout, cleft palate model, and you're able to get an output, pretty similar to our single cell RNA sequencing, where it clusters in an unbiased way, and it gives you, different, you can run differential expression analysis, you can define, region of interest, and you can quantify both cells and, different, mRNA outputs. And what we really found was that in the absence of PAX9, Wnt signaling was really dysregulated across all cell types. And that was intriguing to us because we saw that PAX9 was really restricted to that mesenchymal subcluster. But it seems that, we're also having Wnt signaling aberrations in the epithelium, in extracellular matrix, like in all these other, areas of the palate. So this is truly kind of a global knockout of the PAX9 function. And now we wanted to try to visualize our Wnt antagonist molecules in situ at a better resolution than we could in our single cell analysis. So here we're looking at DKK1 on the top panel and DKK2 on the bottom. And you can, kind of appreciate the type of analysis you can do just head to head with genes with the Xenium software. You can, define your region of interest in the square micrometers. You can look at number of transcripts in each of those areas. You can look at the number of cells to kind of compare, apples to apples. And this was a useful tool for us to do just some very basic, head to head, gene expression analysis. And we did this, overlaid with some bone formation markers such as SP7. And it allowed us to identify exactly where in the palate these upregulations of Wnt antagonist molecules were happening. And here, this was kind of the key finding in this phase of the project, in my opinion, was that, the DKK1 molecule was really restricted within the bone matrix itself. You can see it kind of lateral to the midline cleft. And that's to be expected. DKK1 has been, suggested as an osteocyte marker, like one of the markers of an osteocyte. So you see these kind of, terminally differentiated osteoblasts and osteocytes within this like restricted bone that hasn't been able to grow towards the midline. But DKK2, conversely, is really more expressed along that midline mesenchyme that is not differentiated to the osteoblast lineage as of yet. And given its function as a negative regulator of Wnt signaling, we are hypothesizing that DKK2 could be, given its spatial orientation within the cleft, could be restricting that midline progression of Wnt-driven bone formation, which could be underlying the phenotype. But of course, we're gonna run a bit more. We have to run more functional analysis to really claim that. But I think that this is just one of the many ways that spatial biology can really give you clues into form and function inside the organism of interest. And of course, there were many other signaling pathways that we profiled, but we just you have to graduate at some point. So I put together a bunch of supplementary data of different molecules that were also upregulated and downregulated within this cleft palate model. And I think that there's a lot more that can be studied and should be studied within this context. And now kind of the in progress work that's not published yet. So don't scoop me, please. But this is kind of diving deeper into this cleft palate, spatial transcriptomic world here. So we're wanting to use a bit more of because a lot of this data that I just showed you, we were so early to adapt some of the technologies at the NIH. We were some of the first runs on the Xenium analyzer, the first runs on the Visium, that none of the computational pipelines had really caught up to the bench work yet. So we did a lot of this kind of at a very basic, what we could do in-house type of level. And I'm not a computational biologist by any means, but now that the computational pipelines are increasing, there is a lot more that you can do with your data sets, as you all know. And so now we're kind of, rerunning and reanalyzing and growing our data sets to include more stages of development. And now, combining and integrating data sets to be a bit more impactful, a bit, larger scale. So this is I'm just giving a shout out to our recent summer student, Evan Stepano from Columbia University Dental School, who really, he really paved the way computationally to start doing a bit deeper dive into the spatial transcriptomics of our data sets here. So this is kind of a snapshot of what we're working on now. And I'd love to collaborate with people who are smarter than I am to really make this a reality. But this is one way that we're able to kind of create subregions within our developing palate. So you can see, the way that the Xenium analyzer allows you to bin the expression of different genes within these like tiny squares along your tissue. And we used some known markers that are, clustering along the oral palate rather than the nasal palate. That's the OP and the NP. So the oral palate has particular, biomarkers that the NP does not. So this allows us to kind of, exclude the region-specific anatomy of the palate while we do some more like sub-regional analyses of this tissue. So we started by doing this, and then we grew our data set by including earlier stages of development, day 12 and day 13, both in a wild type situation as well as in a knockout situation. But first to study, normal development at day 12 and day 13 of the palate when it's still growing along the sides of the tongue, we were able to harness a bit more of the power of Xenium using all 350 of these genes and create a bit more of a computational differential expression across all 350 of these genes. And so you get a volcano plot type of output similar to what you might get in a bulk or single cell RNA sequencing. And it allows you to kind of, capture a bit more of the data set that you're generating. And we did this for both the wild type and now for the knockout. We're in process of doing this. And so this is kind of the building of the data set that we're excited about. And you can really validate a lot of these findings, both, quantitatively, but also in the tissue, which is what you want to see. So what we started to find is that in our cleft palate model, we have, disruptions in homeobox genes such as ALX1 and PRX1. You can see the gene expression on the top panel, as well as the density of the gene expression on the bottom panel. And I think it's helpful to see both. You get kind of the raw mRNA expression profile on the top, as well as the computed density of that mRNA, because sometimes it's hard to tell in the two-dimensional plane, where those molecules are located within the cells. And then we also have our Wnt genes that are disrupted, which is not surprising. That's kind of holding true to what we found previously. But also ECM genes are lighting up as very significantly, dysregulated in our cleft palate model, which, as we delve more into more of the Wnt effector functions within this system, we're realizing that Wnt does play a role within the ECM, not only with bone formation and cartilage formation, but also the ECM that's supporting it biomechanically. And so we see several ECM genes, elastin, osteoglycan, and then COL1A1 as well, being disrupted in the absence of PAX9. So these are all valuable hits that we're also validating with RNA-scope, and we're able to really quantitatively and qualitatively kind of map back and see the spatial transcriptomic differences between the normal palate and the PAX9 knockout. And so this is our kind of working hypothesis that PAX9, being a patterning transcription factor, is impacting these homeobox genes during development that then impact Wnt signaling and can directly be associated with these disruptions in ECM formation, which could be an underlying cause of this disrupted and atrophic bone that is failing to form towards the midline of the cleft. And this is just one more viewpoint of this working hypothesis that we're seeing. And kind of the next chapter of this, which is in process now, I was just telling Sean about it. We, to kind of grow the capture of this type of data set, we've recently run Visium HD, which is kind of the next generation of this whole transcriptome spatial RNA sequencing, but at a much higher resolution. We're able to resolve this spatial information at a 2-micron scale, up to a 2-micron scale, which is supposedly a single cell resolution. So we're pretty excited about the output thus far. I don't have that data to show you quite yet, but if anyone is interested, we can chat about it as we continue to analyze it. But that's going to be kind of the exciting discovery pipeline, because I think that as spatial biology evolves in the basic science world, I think that it will continue to find uses in the clinical and translational sciences world, which is really where we want to go next. So we're taking all of these fundamental truths that we're learning from basic developmental biology, and what we'd really like, what I dream about doing as a clinician scientist, is applying translational therapeutic applications in real patients one day. And that's kind of the pinnacle that we all strive for. So we kind of reviewed the state of the art for cleft patients and what type of non-autologous sources of bone formation can they turn to if they're trying to regenerate that bone that has failed to form in the palate. And a lot of them are coming up short in the preclinical phase, and they're not making it to the clinic. And those that do make it to the clinic, they don't quite form ideal structures of soft tissue and hard tissue. And so it's a difficult journey to get there, but I do think that it's worth investigating. I'm going to put one plug out to our recent collaboration article here with Steve Gowdy's lab at Emory, where we really put together multiple different components here, but it's that the goal was to be able to not only regenerate the hard tissue underneath a cleft defect, but a lot of times these patients, after their surgery, because of scarring and fibrosis that forms within the mucosa and the soft tissue overlying that hard tissue, it'll oftentimes degrade, and it'll cause a communication between the nasal and the oral cavity, which is a oral nasal fistula. And to correct this, surgeons oftentimes have to cut out the scar tissue. They have to reapproximate a flap. They have to rearrange tissues. But our groups were able to design a biomaterial that is impregnated with an immunomodulator, which is its function is really to modulate the egress of lymphocytes and other immune cells within that implanted site, to allow for more of a pro-regenerative rather than a pro-inflammatory state. And we were able to show that the soft tissue is able to heal at a much faster rate than if a blank biomaterial scaffold was to be placed in there. And so this is kind of an initial step towards more of an application towards translational interventions that we could be doing for these patients. And we're going to be pulling from some of these principles in the soft tissue regeneration that we were able to achieve in that last project more into the hard tissue now, because these patients oftentimes between the age of 5 and 8 have to undergo a very painful, bone grafting procedure where, the surgeons will take them in and they'll shave off a piece of their iliac crest bone, and they will, shove it up into their upper jaw, and it'll take sometimes it won't. And it's just a very, painful process that might not need to happen if we can come up with something off the shelf that can allow for that bone to heal properly, for their teeth to erupt properly, and for their entire, facial skeleton to grow properly as well. So this is kind of something cooking right now in the lab, where we are, designing off of a PCL backbone, these nanogels that are coated with BMP2, and, hopefully enough to, stimulate a pro-osteogenic environment. And then, we're looking to design an animal model where we could be, creating a very consistent defect within the palate, similar to what a cleft palate or an oronasal fistula might look like, and be able to have a more, regenerative approach to solve this issue. But really, these are kind of the big steps on the horizon for us as we try to learn from our developmental biology experience and apply it in a more, engineering focused and functional way. And I hope that we can, obviate the need for these autologous bone grafts in patients one day. That's kind of the overarching hope, because, cleft and craniofacial care is so complex. There's so many moving parts and so many different disciplines that weigh in. And I think that this is something that needs to be addressed before we can really move forward in the field. But I'd just like to acknowledge the team here of supporters. This is our PI, Dr. D'Souza. And then, I have a really supportive team around me. And of course, at the NIH, you're never short on collaborators or, support systems and cores that allow you to do a lot of great high-powered research. And I'm learning every single day how to just push the needle and do things in a collaborative way. So that's really kind of my call to you all too. I'd love for this to be, the beginning of a relationship where we can share resources. I kind of feel indebted while being at the NIH. We're so blessed with, technology at our fingertips that I'd like to be able to collaborate with many of you if you're willing. So please do reach out to me at any time. This is my contact info, and I'd love to take any questions at this time. Yes, thank you. Thank you. And I'm told I need to repeat the questions for online. So yeah, great question. He's asking if we want to do some sort of functional assays in the lab to assess the genetic interactions between some of these molecules that I described, such as PAX9 and DKK1 and 2. And I didn't feel like I had quite enough time to cram it into the talk, but that is part of what our lab has done already in prior years. Before the era of omics and sequencing and everything, PAX9 had already been, described as a potential, biomarker of cleft palate and tooth-agenesis. And one of the papers that my PI had published, several years before I joined her lab was just the direct interaction of PAX9 to DKK1 and DKK2. So it was shown that it binds to the transcription. One of the ways that PAX9 can orchestrate Wnt signaling. Fairly, since we know there is this genetic interaction between PAX9 and the DKKs. I do think that there are others, though, within the Wnt signaling cascade that are likely, culprits that we could target. Some druggable targets, in fact, such as sclerostin is on my mind, because it's another, surface level, protein that is binding and inhibiting Wnt signaling propagation. So I do think there's a lot more room for, for functional assays, which I haven't focused on yet. So that's on the docket. Please.
Really nice talk.
Thank you.
So one of the things with palates is that you know, it's very heterogeneous disorder, right? So
Yes.
you've got genetic forms, you've got syndromic, non-syndromic forms. So I guess, I don't know if you starting to consider the idea that maybe if you get, maybe there are certain common things, certain common pathways that then leads into various different things, even though there's multiple causes of it. So I don't know if you have any thoughts about that or that.
Thank you for that comment. Yeah, that's on my mind a lot, you know, given, so let me just repeat. So he's asking just about the heterogeneous nature of orofacial clefts, given their origins being so diverse in genetic cause or environmental cause. Yeah, how do you address such a heterogeneous pathology, right? And that's absolutely front of mind for me, and which is why as our, you know, our project has kind of evolved over the years. We kind of initially thought that we could be intervening in more of a preventative fashion to try to, you know, correct the signaling environment in utero where you know, there may be a disruption in certain genetic causes of cleft palate. That could certainly still be an option, but that's probably farther down the road. But what I think could be kind of a common middle ground as we intervene, you know, postnatally, once these children are born and they're initially taken care of, you know, the lip is repaired, the palate is repaired, but they're still missing bone underneath that soft tissue. And that is independent of genetic or environmental cause, right? That's just a morphological truth of a cleft palate, is that, you know, the alveolar bone is lacking in the maxilla. And so I think that could very well be an avenue where we are addressing a clinical need, but we are not tied to a particular, you know, etiology of the cleft. It's not just, you know, certain clefts that have this. It's all clefts of that particular, you know, phenotype. There is a spectrum of phenotypes of orofacial clefts, of course, but any bony cleft we could address perhaps with some sort of regenerative, you know, non-autologous source to help promote wound healing in that sense. So that's kind of where my attention is turning as a biomedical engineer too, and as a clinician. That's something that I think is attainable within, you know, the foreseeable future. I do think that, you know, gene therapies and replacement therapies are maybe an option as people continue to study these things, and there are case scenarios of, you know, congenital anomalies that are corrected in utero, but it's rare. So I think that the bigger bang for our buck, honestly, is to try to target a common, you know, pathology amongst, you know, many clefts, which is, you know, the deficiency of bone within that space. Does that make sense?
Great, thanks so much.
Thank you.
So I have a question. In the first part of your presentation, you mentioned the primary serial molecule involved in the pathogenesis, especially that expressed in the osteogenic.
Mm-hmm.
And the second part of the presentation, you mentioned PAX9 knockout models. The Wnt pathway was disrupted.
Mm-hmm.
Do you have any kind of the link between the primary serial and the Wnt pathway?
Yeah, thank you for the question. Yeah, so he's asking about kind of, yeah, part one of the talk focused on that unexpected finding of the primary cilia playing, you know, likely a potential role in the palatal fusion process. And then the second part of the talk didn't really readdress that. It talked more about, you know, PAX9's, you know, direct function over Wnt signaling, and that, you know, downstream, you know, transformation of the palate phenotype. And I mean, my short answer is that I don't know a direct link between, you know, Wnt signaling and primary cilia function. From what I have talked to some cilia experts within the craniofacial biology field, and they have told me that there's not much evidence of a direct link with you know, with regard to Wnt signaling and you know, primary cilia function. I do think that BMP signaling is a bit more intricately related, and sonic hedgehog signaling, some of these overlapping pathways within, you know, the same, you know, network of signaling cascades, those are more directly impacting primary cilia. But there is a growing body of literature, from what I understand, of, you know, cilia function within, you know, bone formation, that cilia are critical for bone to properly migrate and differentiate. And I'm not an expert in that, but I'd like to learn more about that and how we can maybe couple these findings together. Thank you for that question. It's usually.
Yeah, a great thanks. So it's somehow related to this question. So
Oh, yes.
that the primary function could be mainly after the growth factor or the mechanism sensing. Then the pathway you think play more of the palate fusion.
Growth factors or mechanosensing. Yeah. Okay. Yeah. So the question is, which one do I think plays a more prominent role in the palatal fusion event? That's a debatable question. You know, I feel like a lot of people will have different responses to that. You know, I am probably just going to take a balk at that answer and say that it's definitely both. I think that I have a particular bias because we focus so much on, you know, signaling and growth factor, you know, in our lab. We're very molecularly driven. But if I talk to a mechanobiologist down the hall, they'll probably say the complete opposite. But I know that it's both. It's definitely both. I think that there's a lot that is still to be understood about that fusion event in terms of that epithelial seam that I know Jeff Bush at UCSF has done some really beautiful work of, yeah, and Rulong Jiang as well, of you know, watching those cells in real time either migrate or, you know, undergo epithelial to mesenchymal transformation or undergo apoptosis. Like there are probably multiple, you know, pathways in play at that moment of fusion that's allowing for that mesenchyme to become confluent and fuse as soft tissue. I think it's both. Is my short answer.
Yeah. So yeah. Then if I understood correctly, so the posterior portion of the palate, the more the mechanobiology related mechanism to raise and fuse. So that it's just called. So you will be able to differentially look at the anterior part and posterior part. Give us some idea which the differential regulation, differential involvement function in the country. I think that's a great advantage in the system.
Thank you. Yes. He's just making the statement that there are key differences between the anterior and the posterior parts of the palate functionally, and that if we were to, you know, differentially analyze these, we may be able to, you know, understand better this question of functional differences. And I agree. I think that there's some really elegant, you know, literature that shows kind of the sequence of how those palatal shelves initially touch. And I want to say it's really the middlemost portion of the palate that initially touches with the epithelial edges, and then it almost closes like a zipper in both directions. So it's really an intricate process that, yeah, has so much to do with mechanobiology. And I wish I were an expert in all of that, but I need to learn more about how those cells and actin filaments are interacting to allow for those cells to interact. Yeah, thank you for that comment. I agree. Thank you.
I'm not an expert in craniofacial. That's not the tissue I study. So I was more wondering into the computational aspect of the interaction between cells. So in the tissue that I study, which is the colon, the cells that react to wind are not the producers of it. So there's a lot of connectivity and cell interactions with all the other cells around, which are mainly the fibroblasts in the tissue that are sharing to the epithelium. And I know that there have been some updates to some packages like Cell Chat and Cell Phone DB that are starting to be able to be used spatially to start asking this question. And because you have a bias between the front of your tissue being DKK2 and the back being DKK1, I don't know if you have tried using these type of packages to try to see the interaction between one of them and seeing if that's one of the reasons why they're also like having a different expression. So if you're taking DKK, that could be, I don't know, give you some idea of what could be.
I love that. Thank you for that comment. Yeah, just to repeat for the online folks, he's bringing up some of these, you know, more, you know, elegant computational pipelines that are available nowadays, such as, you know, Cell Chat, which we have used in other studies in our lab, and I do like it. I think it allows you kind of a deeper level of cell-cell interaction understanding when it comes to some of these outputs of single cell RNA sequencing. I actually haven't used it in a spatial context yet, but I think that would be really valuable. I agree, to really delve into why certain, molecules of interest are, expressed on the midline portion versus the lateral portion. I think that could be a wonderful adjunct. Thank you. Please.
First one is about the periderm, and if you sort of like looked at the role of the periderm in your Pax9 mutants, or whether or not like any of the spatial analyses has been able to identify like a marker for the periderm. It's kind of an up-and-coming tissue in like OMC pathogenesis. And then
Yes.
The second question would be, I think about like Terence Capellini, for example, is doing a lot of work with like human fetal long bones and doing a lot of spatial analyses of bones. So do you think that like there's a time when we'll be able to get human like embryonic tissues and be able to do spatial like CNIOM and CDMHD in those tissues?
Oh, thank you, Sean. Great question. Sean projects well, so maybe you all heard him, but his first question was about the periderm, which is kind of a niche cell population that has gained some traction in our field lately. It turns out it's like a very functionally significant cell population beyond just the epithelium or the mesenchyme. And I'll admit that I've been kind of tunnel visioned into our mesenchymal cell populations, given the functional role of Pax9 that we're pretty familiar with, and given that I'm studying primarily the bone of this tissue, I've really focused much more on the mesenchyme than other tissues or other cell types. And I do think that I've gotten this question from someone who's a periderm expert, and they're in the process of helping me to kind of understand our clusters, because there is a cluster of cells from our single cell RNA sequencing of the palate that we can definitively say is the periderm. I'd like to understand if Pax9 has an upstream effect on the periderm and its potential extension or lack of extension in a cleft palate type of situation. Have not yet, so that's definitely on the horizon. But your other question about perhaps having access to the human fetal specimens of the craniofacial complex with the advent of these other types of tissues being available nowadays, I do think that's very plausible. I think that especially at the NIH, that seems like the type of place that could maybe push the needle on this type of protocol. I'll have to ask some people higher up before I can really answer how feasible that would be. But I think that that's something that is kind of next step for us to like take this technology a bit more translational and to try to understand how it could actually impact clinical practice rather than just kind of a playground for basic research. And so that's something that I'd really like to try to push forward in the next few years. Thank you for that question.